This lecture will be the first in a series where we consider the properties of tissues. As I mentioned in the introduction, physiology is concerned with multiple levels of organization. And our initial focus is going to be on the tissue level. So below that, of course, we have cells, and below cells, we have molecules. And so a lot of what we will rely on to understand what happens at the tissue level is the dynamics at the cellular and molecular levels. So, what do I mean by tissues, or what are tissues? We actually have just four types of tissues in the body. And the type that I'm going to talk about today are connective tissues. These are largely or entirely extracellular structures that can be maintained by cells within that extracellular structure. They include the attachments on the ends of muscles, which is what we have highlighted here with this tendon up top. But we'll see lots of other examples of connective tissues. And connective tissues, largely for the rest of this course, play a supporting role. So in this lecture, they're the focus, but we're going to mostly mention connective tissues in how they influence the other types of tissues that we're going to be focused on. And those include epithelial tissues. Epithelial tissues provide a barrier between the interior of your body and all of its physiological functions and the outer world. They also mediate the exchange of molecules, the taking up of nutrients in the digestive system, the production of urine, for example. We have nervous tissues, so neurons, and there are various higher levels of network structure. And then finally, muscles. So that's it. We're just four types of tissues. And we're going to consider them across lectures in sequence, starting with the connective tissue. All right, so what are the functions of connective tissues? I've described that you have them in the case of tendons, but there are a wide variety of functions. And so here we have our schematic for the physiology of the body. And largely when we're referencing these organ systems that are shown here, we tend to think of the body as being this kind of fluid sac that is encased in epithelial cells that provide a barrier for this fluid interior environment. And we're largely focused on cells and molecules and how they're kind of bounded within the body. But when you're thinking about connective tissues, it's helpful to think about the structure of the body. Okay, so instead of us just being this liquid sac, we actually have bones, tendons, ligaments that attach muscles to a skeleton. And when those muscles generate force, they animate the body. And so it's in this context that we can consider these major mechanical functions of connective tissues. So we have, for example, a femur, a bone, under load. And in that situation, what does the bone provide? Well, it's stiff and it's strong, but yet it's relatively lightweight. We have tendons, as I've mentioned, which transmit the forces generated by muscles. Surrounding your lungs, there are connective tissues that provide an essential recoil to the inflation of the lungs as you inspire air. And that recoil provides a passive return of elastic energy onto the lungs, which then supplements the action of the surrounding skeletal muscles to have expiration. The gut wall has an incredible ability to change shape, largely by reconfiguring the connective tissue that lines it. Finally, when we talk about the circulatory system, we're going to see how the connective tissue around the major blood vessels, in particular the large arteries, are essential for, also similar to the lungs, recoiling, storing elastic energy, returning to their original shape as those arteries become pressurized and relax on each heartbeat. And then finally, we have connective tissue that's not just playing a role in the transmission of forces, but it provides protective functions in, say, for example, padding the organs, which cushions the kidneys, which are very delicate structures, and, for example, your eyes against impact. So, in light of these major functions, I'm sure there are plenty of others that we could enumerate as well. This just gives you a sense, an overview of the major mechanical functions of connective tissues. For this lecture, we want to address the question of how tissues vary in their mechanical properties. We have bones, which are stiff and strong. We have tendons. We have connective tissues around the arteries, around the gut wall. They're intended to deform, unlike bones, and to store energy elastically and to return that energy. So we have disparate mechanical needs for these different functions. How is it that experimentalists can characterize these mechanical properties, and what are the inherent properties at lower levels of organization? We're talking molecules and cells. What is it about these lower levels of organization that endow these tissues with those different mechanical properties? And this is a large field of research, just like every topic that we talk about in this course, where there are whole fields that are concerned with trying to understand how the molecular composition of tissues influences their mechanical properties. So we're just going to scratch the surface. Here we have a schematic of the leg of a horse, and we've highlighted in the orangish color here tendons that connect muscles, wrap around joints, and then apply the force that is generated by the muscles on an insertion on a bone. And we're going to contrast the anatomy of the horse with what we might see in a mouse. And I'm scaling up the schematic of a mouse, but of course a mouse is much smaller. You can see the mouse has this crouched posture, but there's a similar arrangement of its ligaments in its legs. And what we want to do is to contrast the mechanical properties of the tendons in these two species. Now, the way that scientists often probe the material properties and the structural properties of a piece of tissue such as this is they'll essentially attach one end of this tissue. So here we have the tendon that's suspended from an anchor point, and on the other end, we're applying a force. And I've shown it as if it's a weight that we're hanging off the end, which you could do in a very simple test. But there are more sophisticated ways of doing this as well, where you have a force probe and a motorized setup so that you pull on the material with a known force. And in this context, since we've essentially stapled our piece of tissue to a ceiling and we're pulling on it, then we can infer that the material inside or in between where it's being attached at both ends is being pulled, and we therefore say that it's placed in tension. And under these conditions, we can track how the tissue changes its length as a function of the force that we apply on the end of our sample. And what we have here is a graph of those measurements. And note here that this is the change in length. So we don't start with a zero length, but we start at a zero change in length. So we take this piece of tissue, and its initial length at rest is shown here. And so the change in length is how much we're extending the tissue beyond that resting length. Now, for a horse, we extract the tendon from the cadaver of a horse. we apply different forces to it, and we measure the change in length. And the convention is to have force on the y-axis here, even though the forces essentially are independent variable. But if you do that mechanical test, then you'll find a curve that looks something like this. You do the same for the mouse tendon, and you get a curve that looks like this. And at the ends of these curves, we have failure. That is a breaking up of the tendon, and therefore the inability to measure any further forces in both cases. And you can see that the horse tendon achieves a much higher force before it breaks. So we call that the breaking force, than the mouse tendon. And the question here is like, well, why are they different exactly? Why does it take more force to break the horse tendon? And quite simply, the answer to that question, well, theoretically, it could be that the mouse tendon is composed of different material, or it could just be a function of the fact that the horse is really big and its tendons are therefore much larger than the mouse tendons. And therefore, maybe it's actually a function of its size. And so the way that scientists in this area will distinguish those two categories of possibilities is that they'll refer to the composition of the tendon as material. And material has certain properties that can be distinguished from structure. And the size difference between the horse and the mouse is one way of thinking about structure, that structure is scale dependent, whereas material properties are inherent to the material and therefore not scale dependent. So the mechanics of an organ depends on both its structural properties and the composition of the material that creates that structure. So, how do material scientists distinguish between structural properties and material properties? In the measurement that I just outlined, we're not making that distinction. But what we can do is we can take those measurements and we can transform them. So, on the left, we have the measurements we described, both the material composition and the structure of the tendons are going to influence those curves. We can remove the structural aspects of this. The force that we're applying to our material acts on a cross section of that sample. And you can just imagine a population of molecules all being stretched as a consequence of the force that we've applied on the end. And what matters here is not the length of the material, because the forces acting on any particular molecule are not altered by having a long tendon, but they are altered by the number of other molecules that are working, that are under load on the cross section of that material. And so if we want to sort of remove the structural aspects of the loading on those molecules, what we would do is we'd consider the cross-sectional area of that material. And if you take the force that's applied and you divide it by the cross-sectional area, then you're effectively normalizing for differences in cross-sectional area. And therefore, you're getting at how the molecules are being loaded. And that force divided by cross-sectional area, which we see in the graph on the right, is known as the stress. And it has units of Pascal, just like pressure. And you can think of that as stress. You can think of stress as being, how much molecules are being loaded in any particular structure, such as this. What about length? Well, here, the initial length does really matter. If you have a super long tendon and you hang the weight on the end of it, then it's going to tend to stretch more than if you had a really short tendon. And so, what we can do with this change in length that we measure in response to loading is to divide it by the initial length of the sample. That's the way that we can normalize for differences in samples. Let's just say you grab tendons from the horse, you grab tendons from the mouse, and we can correct for the size differences between them by doing that transformation there, just dividing the change in length by the initial length. We call that strain. So stress and strain are often treated as synonyms in popular language. For mechanical engineers, material scientists, they're very distinct quantities, where stress is referring to the force per unit area, but you're thinking in terms of forces. And then strains are what occur in response to those loads. So how much is it deforming is a reflection of its strain. Now, when you take the two curves that we measured on the left for the horse and the mouse, and you do these transformations, divide by area, divide by initial length, then what you would find is that the two curves would actually be right on top of each other. And this is because the tendon in the horse and the mouse have the same material properties. And so once we have removed the influence of structure from the differences in size between the two animals, then we are revealing this consistency in their material properties. So stress and strain are the key to revealing the material properties of any particular biomaterial. Now, along the way, we mentioned this property where the material breaks. That's known as the strength. So again, we often in popular language will refer to strength, stress, strain as being very sort of similar terms. Here they're very different. Strength refers to a particular either force or stress. So the strength of a material is given as its stress. The strength of a structure and material would be given as a force. Another key property in this relationship between stress and strain that we see is given by the slope of the curve. And because it's a nonlinear relationship, that slope changes. But it's typical, if you have a nonlinear curve like this, to measure the slope in the middle. And we call that the stiffness. In this case, it'd be the material stiffness of the structure. Again, if you are interested in the the material stiffness is it's characterizing the stiffness of the material. You could also measure stiffness in terms of the force as a function of length changes, and that measure of stiffness would be reflecting both the structural and material properties. But since we're focused on materials here, we'll just go with the term stiffness to refer to a material property. And again, as we laid out at the outset, we have different mechanical functions in the body, and therefore different material properties to match those mechanical demands. So let's get some examples here. Here we have a table with different materials, different stiffnesses, so the slope in the middle of that line, and different stress values where they break, known as the strength. So we've been talking about tendons. We have these particular values. They're not all that interesting until you compare them to other things. So for example, the fat tissue that helps insulate your kidneys and your eyes from mechanical forces, that fat tissue has a much lower stiffness than tendons. Perhaps not surprisingly, because you think of fat, those of us who eat animals are familiar with fat tissue. It might have some structural integrity to it, but it's a whole lot easier to bite through if you're eating than is tendon. And in fact, this is a 300,000 times difference in magnitude. So quite often, meaningful stiffness differences vary by orders of magnitude. Cartilage is a tissue that provides a shock absorber between your intervertebral joints, that is the bones in your spine. And we have that at 10 megapascals, and it has a strength of 5 megapascals. And this is a 20-fold difference from the strength that we have in tendon. Finally, bone exceeds the strength of tendon by at least a little bit, and it has a higher stiffness than what you see for cartilage, which is perhaps not surprising, much higher. Now, all of these values correspond to the kind of mechanical test that we've laid out so far. That is, we're pulling on the material, and so not surprisingly, we don't even have a value for the strength of the fat tissue because it really doesn't resist pulling very well at all. And cartilage allows for some resistance to pulling, same with bone. But those tissues do quite a bit better when you take your bit of material and instead of pulling on it, you squish it. So you apply a load at one end, and what we're doing here is we're taking our tissue and we're compressing it. And in compression, you can get very different properties for your material. So for example, in bone, we can see that it's stronger in compression than it is in tension. So materials can have differential responses to the direction of loading. And more broadly, we can say that materials differ in their composition to allow for these differences in material properties. And I'm just going to march through a few examples because what it's going to allow us to do is to itemize some of the important features of the materials that help inform these differences in their properties. So for example, we have tendon. What we're looking at here is a histological section. And you'll notice that there are these pink squiggly bits here that are highlighted in this histological section. And what those are collagen fibers. So collagen is an elastic protein that plays a critical role in determining the mechanical properties of any particular material. And just like a rubber band, collagen fibers can get really stiff if you extend them to great lengths. But they're really floppy in compression. And so they don't help a tendon much when you try to compress a sample of tendon. But what you do find with this material, tendon, is that despite the fact that most of what we're looking at here is extracellular, that there are supporting cells that are located in this matrix of collagen fibers. And they break down the material and they construct more material. They're constantly remodeling tendons. Tendons will become much more robust under loading. So if you do strength training, then your tendons are going to tend to be stronger by virtue of the work done by these cells. And they will, if you're in, say, a reduced gravity environment or you have a sedentary lifestyle, those cells will break down the tendon accordingly because these materials, these tissues require energy to stay supported. And the collagen fibers are a source of energy. Your body tends to operate relatively efficiently. And so what it will tend to do is to break down extracellular structures that are not really being used. Okay, so that's tendons. So tendons are dominated by the presence of these collagen fibers. For our weaker adipose tissue, okay, so adipose tissue is a way that, or it's a type of tissue that we use for storing energy. We'll get into its essential role in energy balance later on in this course. But it also has these mechanical functions. They protect organs in the body, like the eyes and the kidneys. And if you look through a histological section of adipose tissue, you will see these thicker bits, which correspond to the nuclei of cells. But most of what we're seeing here is fat. We're looking at lipids that are bound together by collagen fibers. So even in adipose tissue, despite its relatively weak material strength, does have collagen fibers so that it has some structural integrity. Next up is cartilage. Okay, so cartilage plays an essential role in shock absorption. And what we have here is collagen fibers that are surrounded by what we call ground substance. So ground substance generally corresponds to a proteoglycan hydrogel that resists compression and it dampens, but it can't resist any tension. So if you pull on this gel, like Jello, if you were to pull on Jello, then it sort of breaks apart. It fractures really easily. But what the collagen fibers do to help on that front is to offer some structural integrity to the cartilage. And again, we have cells that produce and they maintain the matrix. Now, there's another type of ground substance. So we changed the language here to often a proteoglycan hydrogel. Ground substance can also be found in bone, where it's not a proteoglycan hydrogel, but instead it's composed of hydroxyapatite crystals that fill spaces within a collagen scaffold. So bones offer kind of a special case of ground substance. You still have cells within your bones that are breaking down and rebuilding this stiff material. And it makes sense that bone would be sort of a special case because it is so stiff and it is relatively strong. And it does that through the hydroxyapatite crystals. Okay, so there are some overriding patterns as you look across different types of connective tissue. And let's just try to summarize that. Okay, so I mentioned the essential role played by collagen fibers that are like rubber bands at a molecular level. And then we also have this ground substance. And then in addition, there are these support cells. So together, the ground substance and the collagen fibers compose what is referred to as the matrix. And so we started with tendon, but we have a similar composition for cartilage, where we have these supporting cells that are suspended in the matrix, having the ground substance and the collagen fibers. There's another type of tissue we haven't yet mentioned called loose connective tissue, which is basically the same kinds of parts, but fewer collagen fibers, and therefore a less stiff material composition and tension. So this can be a soft liquid or a gel that's reinforced by those fibers. Again, tendon has just a higher composition of those collagen fibers, and they tend to be aligned more so than what you have in the case of the cartilage because the loading is predictable and those collagen fibers are therefore aligned with the direction of tension that's generated by the muscles to which the tendon is attached to. Ligaments are very similar to tendons, but they connect bone to bone and they help stabilize joints. They play essential roles in sort of keeping your skeleton together. Your skeleton, of course, is composed of bones. And for bones, we have this unusual ground substance of using the hydroxyapatite crystals instead of a proteoglycan gel. So these are all structural connective tissues. We're going to, again, refer to them lots and lots in the course, but there are also physiological connective tissues. So actually blood qualifies as a connective tissue. Its ground substance is liquid. Okay, we're talking about plasma there instead of the proteoglycan gel, so kind of a special case, as are the adipose tissue that compose the fat stores in your body, which we did talk about. All right, so the question for this lecture was, how do tissues vary in their mechanics? We have this toolkit of doing materials testing to separate structural and material properties. This, by the way, should be a change in length on the x-axis in these two graphs. There's a little typo there. We have the direction of loading being an important distinguisher between different types of material properties. And those material properties are a reflection of the material composition of that tissue.